第69回日本生殖医学会学術講演会総会株式会社リプロライフ共催セミナーを開催いたします。本日のテーマは CAPA-IVM IVM 臨床応用拡大のキーポイント Basics and Clinical Aspect of Capacitation Ivm になります。本日の座長を医療法人昌永会 IVF 大阪クリニック院長の福田愛作先生にお願いしております。それでは福田先生、よろしくお願いいたします。
おはようございます。今日は IVM ということで、そんなに多くの参加者じゃないんですけれども、IVM のコアな支持者がおられると思いますので、楽しみにしていただきたいと思います。It's my great pleasure to have the two guest speakers, and JOHAN and also TUONG, both long time my friend, and I'll speak in Japanese from now. それでですね、今日は2人の演者の先生方です。で、最初はですね、ヨハン・シュミッツ、ベルギーのヨハン先生ですけれども、彼はですね、もうずっと、ヨハン、プリス、ずっとIBM されてまして、彼は僕の師匠、he is my mentor of the IBM and long time friend で、ずっとIBM をされております。で、今日はですね、CAPA-IVM、いわゆる2段階 IBM についてですね、その、基礎、彼は MD なんですけども、基礎をずっとされておりまして、今、世界で CAPA-IVM が広がりつつあります。で、日本はちょっと保険とかでですね、最近 IBM される施設減ってるんですけども、今日の話を聞かれてですね、また IBM をされる方が増えると思います。Johan, please start your lecture.
Thank you, Mr. Chairman. Dear Isaac, thank you for translating the things for me for many years. Dear colleagues, first of all, I'm very honored to be invited here by RiproLife to talk about IVM and more specifically about CAPA-IVM. CAPA stands for Capacitation Ivm, and the first 20 minutes are dedicated to the way we developed it and why we think that CAPA-IVM is a necessity to have good quality embryos. So, as you can see here, the CAPA system goes from pickup over pre-IVM to IVM, and wouldn't it be great if we would have a system where the patient comes into the clinic, she has an ultrasound and an AMH measurement, and you see that the AMH is over 5, and that you can see many follicles, and that immediately, without any pre-treatment, you can do the pickup in the patient. But then, of course, there are conditions, and the conditions are that we can culture these very compact oocyte cumulus complexes that you can see. I'm trying to find the pointer. Yeah, here. I'm trying to push the red button. Yeah, that you can see here that the oocyte cumulus complexes are very dense, so unexpanded. It's not like after ACG. Because our system does not use any LH or ACG. Then we culture it in pre-IVM, and you see it stays, there is proliferation, and then you have only after IVM, you have expanded oocyte cumuluses, then you use the oocytes, you do normal fertilization or you do injection, mostly microinjection, and then you have blastocysts. So the patient doesn't have any pre-treatment, she can go home, we make the blastocyst, we freeze everything, and then the patient can come back whenever she wants to have a blastocyst and a pregnancy. So this is my disclosure. I'm co-founder and CSO of Lavima Fertility, which is a spin-off company of my university, which is VUB, and we developed CAPA-IVM at that university, and we created a spin-off to bring it to the market. So I want to review for you the journey to CAPA-IVM system. And I think the aim, what is the aim? The aim is to use oocyte from small, healthy follicles at any moment of the cycle, preferably without hormonal treatment. This is happening already in most of the animal species. Yeah? They are not stimulated whatsoever. It's only the woman that is stimulated. Is that not surprising? If there is no need, I think it would be great to have such a system. What is the task that we have is point 2. We have to replace FSH and ACG dependent stimulation by a defined in vitro system and prove the biological effects. And that's what we did in animal model, especially we used the mouse model to start and the human model. Then we have to validate that this is a system that is effective and safe in human. That's what we did. And finally, apply it in the clinic, and that's what my colleague Dr. TUONG will do, because it's with him that we developed that, and with his wife, Dr. Lan, in Vietnam, in Ho Chi Minh, and also we did the same here in Osaka with our president and with Dr. Morimoto. So you all know what is IVF, and I show here the cycle of Goujon. Here it's a lot of work to get the large follicles. We have to stimulate, we have to monitor, we have to avoid the risks. But actually, the funny thing is that you have always, in every woman, at any day of the cycle, these small follicles that are present here in class 4 and 5, and these have a very low atresia rate. You see here the word atresia means that the follicles are starting to die off. Yeah? So if the follicle is not dying because you have low atresia, we think that the oocyte is of a better quality. So if you wait too long and the follicles become too big, also there is more atresia and the oocyte is not so good. So if you can catch these oocytes together with the cumulus cells, and I explain why with the cumulus cells, we can make these oocytes mature for IVM. And this is showing the follicle, and you know that the development from the primordial follicle up to ovulatory follicle is a very long journey, and we concentrate with class 5 and 4 and 5 follicles, we concentrate here in this green area. And what you know is that the oocyte grows in close connection with the cumulus cells and with the granulosa cells. And here you see that the follicle is becoming a small antral follicle, so of diameter of 2 to 5, 6 millimeter follicle. And you know that part of these follicles are still blocked in meiosis and are still meiotically incompetent, means that they cannot fulfill their nuclear maturation. But from a certain diameter, a bit larger, they are capable of resuming meiosis. So if you take the eggs out of those classes of follicles, some will start their nuclear maturation, but others not. Now, how can we study that? We have at our disposition many ways of doing that. And you see here in the bottom with Höchst, you see that this oocyte here, the chromatin, is still dispersed, dispersed chromatin, while here, a bit larger follicle, you see that it is surrounded nucleolus, you see like the ring formation, so there is condensation of the chromatin, and then this condensation will lead to transcriptional, so this is what we call the chromatin remodeling, and it will lead to transcriptional silencing. Yeah? So every egg becomes, in the beginning, is full of transcription because a lot of proteins that need to be made for the oocyte growth, and also a stock. There needs to be a stock of RNA particles for the early stages of embryogenesis. And this should be ready at a certain point. But you see here, transcription is silencing, and only if this is the case, the oocyte can go in the new program of fertilization. Now, when you take out the follicles at this stage, you see that there is normally a maturation that happens that takes 10 days more, where the cytoplasm needs to become mature. So if you have the nucleus maturing, but the cytoplasm is not mature, you can expect aneuploidies and abnormalities in chromosome segregation, etc. So you see that many things need to be fulfilled to have a normal embryo, and we are there at a critical time point. So you can have a nuclear mature oocyte, doesn't mean that it can be making a normal embryo. So Dr. Conti and Franciosi in Human Reproduction Update published a very nice paper showing what is needed. You have the cumulus cells here in the green cells attached to the oocyte, and so it is showing all the factors that are important. So there is an integration of cumulus oocyte functions via local feedback mechanisms targeting maternal mRNA translation in the oocyte. And you have the regulation of this maternal mRNA translation in the oocyte. It plays a very critical role in making the normal protein secretion, and there is a feedback between oocyte and cumulus cells. So we call that the bidirectional exchange of signals between oocyte and the surrounding somatic cells. And the signals from the somatic cells stimulate the fully grown oocytes to translate selective mRNAs, and in turn the oocyte secretion factors may control the cumulus cell function. So the oocyte will, under the influence of the cumulus cells, make oocyte secretion factors, and you see them here. It's OSF, it's GDF9, BMP15, FGF2, etc. They have an influence on the cumulus cells, and they ask the cumulus cells to send some metabolites to the oocyte to become fully mature. And then you have here, of course, the somatic cells, the granulosa cells, where normally you have here in blue LH receptor expression. They will at a certain point start to produce amphiregulin, is an EGF-like factor, and you see that on the cumulus cells there is no LH receptor, there is only FSH receptor. But after a certain time there is EGF receptor, and the EGF receptor will bind the amphiregulin and some other EGF-like factors, but amphiregulin is the most important one. And so if you don't have expressed this EGF receptor, you cannot turn on the activity of the amphiregulin on the cumulus cells that will lead to the nuclear maturation. So you see there that we have a little organ where we can, if we are clever enough to put things in the media, that we can turn on the system of cytoplasmic maturation that is normally happening in vivo in a patient. So these are the mechanisms. So the competences in the oocyte are dependent on stage-specific expression of growth factors. I tell you at a certain point you have in the small follicle no EGF receptor. After a certain time the EGF receptor is there under the influence of FSH, and also it's a kind of auto-amplification system, because also amphiregulin is going to be produced from a certain stage on. So if we remove the cumulus cell from a small, what I call predominant follicle, not all the competences are there, and so we have to supplement these factors in the media. And so while in a traditional IVM system you just give it water and some salts, I think to have a better oocyte quality, starting from such a small follicle, you have to give it some growth factors also. And that's what is actually new. And the cumulus oocyte entity has the potential to be self-sufficient if triggered with these growth factors. And so finally, this oocyte that then can lead to a better embryo quality. And what is needed for that? Well, you need to have the transzonal projections in presence. So you see here, cumulus cells, oocyte, and here you have the transzonal projection. It's TZP in abbreviation. Why are these important? Because the cumulus cells are nurturing oocyte growth. You have the reorganization of all the organelles. You have the transcription that is happening there. And this is all directed by the cumulus cells to the oocyte. But of course, you need to have the tunnels there, okay? If you don't have the tunnels, the things are not working. And so making a good culture system means that you need to have these TZPs present. And I will illustrate that a bit further. So why today is IVM not yet in the market? And why is it in all animal species? Because today in the IVM that is commercialized, there is poor embryo quality due to oocyte immaturity. The oocytes have a smaller diameter. The organelles are yet not in the final position. The oocytes are still transcriptionally active. And the cumulus cells did not yet express fully the EGF receptor system. There is no strong connectivity with the oocyte via the TZPs. And so finally, it leads to poor embryo quality. And we hope then with CAPA that it gives us better embryo quality. And this is just to say about the EGF receptor system. It's a publication from Sugimura who showed that with PCR that indeed in a small follicle you don't have the presence of the factors, but when the follicle is a bit bigger and when it has been stimulated for higher cyclic AMP, you have these factors present. Now, the task replace FSH and ACG stimulation by a defined system. We started to use this in a mouse system because this is our usual model. So we take a very juvenile mice, we have the GV oocyte cumulus complex, and what we do first is we want all the oocyte cumulus complexes to be in the same stage of maturation. Because if you have heterogeneity, you have the polar body there at different moments, it becomes a problem for microinjection. So everything needs first to be blocked, and we use meiotic inhibitors. You can do that with cyclic GMP and cyclic AMP analogs or phosphodiesterase inhibitors. And we started to work with these phosphodiesterase inhibitors. So you block it 24 hours, it is still GV, and then only you stimulate it, because in that time you add FSH in the medium, estradiol, GDF9, insulin, etc., the growth factors, okay? So this period, 24 or 48 hours, and then you mature, you do the IVM, like happens today, but is not giving you good metaphase 2 quality eggs. So there were different ways, like you can use, here is the oocyte, you can use PDE3 inhibitors, that's what we did in the beginning, like milrinone or silostamide. These molecules you will find a lot of literature in present, but there was a Chinese student, Dr. Zhang here, Meia Zhang, and also the group from Luciano that used it in bovine. They said, actually it's Zhang that found that C-natriuretic peptide is a small peptide, is produced by the granulosa cells, binds to the receptor on the cumulus cells, on the NPR2 receptor. This activates cyclic GMP. Cyclic GMP goes into the oocyte and blocks the degradation of cyclic AMP. So it blocks the PDE3A, the specific esterase that will degrade cyclic AMP and that will cause normally maturation of the oocyte. So you can block it by having CNP. And this is the natural arrestor. So we don't need to make a culture system with pharmacological compounds. We preferably use what is present already in a follicle, and that can be purified or synthesized because it's a peptide. So this is the system then that we put in the CAPA. It's CNP first, and you see in the mouse what was the result. Here you see the difference. If we take and we do all type IVM, the results are very low in terms of blastocyst. And this is the in vivo control, so in vivo 80%. And then you see that if we use the CAPA system here with CNP and estradiol, with FSH and with GDF9, you see that the results become similar to in vivo. And this is in mice that were not stimulated. So first time when my student Romero came, he showed me the results. I say, wow, this is an enormous positive effect. We have to use that also in human. And so what we saw in the sections before we use PDE3 inhibitor, look here, the oocyte, you see in the zona pellucida is black. There is no transzonal projection. If you use CNP, it's full of transzonal projection. So this shows that CNP and via cyclic GMP is doing something very specific to the transzonal projection. That's why we continue to use CNP. So I think that CAPA-IVM is starting with the small follicles here. This is the classical IVM generally takes oocyte cumulus complexes from larger follicles, but that's not so convenient for the patient because generally she gets a couple of days of stimulation to get larger follicles. So the target diameter should be smaller, 2 to 6 millimeter is more than enough, but then you need this capacitation system. So the idea is then to develop that for the woman. And what is the ideal cohort? Like I say, it is from 2 to 8 millimeter. That's what we did in Brussels and in Vietnam and in Horak. And you see that we put in first a pre-maturation or capacitation culture for 22 hours, 22 to 24, and then only we go into an induced IVM system. And you see that initially we give few days of FSH, only 2 days of FSH, but today for patients with a lot of follicles, it doesn't need FSH, we have just a publication in fertility and sterility. Of course, the first task of a scientist is always to look what do you have present in that cohort. So look to oocyte diameter, transcription, chromatin configuration, to the transzonal projection, to the mitochondrial distribution. And you see here, Flor Sanchez is the other student in 2015 published these data in human reproduction. And here is just an illustration. You see immature oocyte, dispersed chromatin. Here, mature oocyte. Here you see the transcription in an oocyte is still immature. Here it's gone. And here you see the mitochondria. Mitochondria here immature, cytoplasm immature. Here it's getting mature. So you have the methods to see that. Flor published that with the cartoon here to explain you better how it should look. And here you have those data. You see that the oocytes are almost fully grayed, the diameter in these small follicles. Still they have to go a couple of micrometers to become fully qualitative. You see here the transcription goes down in function of the diameter. So we have very well characterized the population of oocytes, and you see that nearly half of the oocytes in that follicle class are fully surrounded nucleolus, and it are only these that can form you normal embryos. So these here need to benefit from your cytoplasmic maturation to get better quality at the end. So I think the capacitation step specifically affects the COC, the cumulus oocyte complexes from 2 to 6 millimeter follicles. And I explained to you already why this is, because the oocyte has still a smaller diameter. There are not the organelles in the good position. Oocytes are still transcriptionally inactive, active, and they should be inactivated, etc. And also no EGF receptor system is there. So we made then the system for human, exactly the same copy-paste as we did for the mouse. So the pre-IVM or capacitation is the same. We use CNP because of the transzonal projections. And we checked that also in the human. And then after 24 hours, having this medium added in a good basal medium, you add also amphiregulin and you have maturation happening. And so how is it effective and is it safe are fair questions. And here you see the system where we do the pickup from the small follicles. First, immediately in that medium, when we do the pickup, we add CNP and estradiol. Estradiol is needed because otherwise you don't have the CNP receptor expressed. So people that don't add estradiol and they say that it's not working is because they have to add estradiol, otherwise you have no receptor. So everything is logic, but you just have to know it. Then here, again, you keep CNP for 24 hours, and then you add only amphiregulin. And this is all material that we have published. Everything is in publication. So the first data in human in Brussels show immediately that here you have 100 oocytes in each group. This is the old IVM, this is the new Kappa-IVM, and you see that the new IVM gives you more blastocysts, at least 2 times to 3 times more blastocysts. See that 24 hours capacitation, you see here, if you look clearly in through the zona, the transzonal projections, also 48 hours gives you nice transzonal projections, and this is important to have the nurturing of the oocyte. It makes our capacitation treatment very simple. Some patients need a pre-treatment because they have a cycle and you want to work at a certain day of the cycle, so you give some oral contraceptive, you stop the oral contraceptive, and then if she is PCO, she does not need any FSH, but if she has a cycle, you give 2 to 3 days of FSH, then you do the pickup. And the kit that we are going to provide will provide all the media and the hormones that we can add in these basal media to do the full capacitation and IVM system. And then the rest is normal media that can be used from the commercially available media. So this is the results that we got between old IVM and new Kappa-IVM, and you see that 3 different publications from 3 different studies, more clinical pregnancies and higher live birth rate. So the application will be given by Dr. TUONG from Vietnam, but I just want to look to the future. And I think when we go to smaller and smaller follicles, what we do is we will, of course, eliminate more and more the use of hormones, but the culture system will become more and more complicated. We just have to know that. But there are solutions to work in lab. It's not that you need hundreds of people. With automation in the future, these things also will become more simplified. So what is the future? I think now we have oocyte secretion factors, and we have also at our availability super GDF9. This is actually a dimer that is formed, GDF9 and BMP15, when they are secreted by the oocyte, they will stimulate the cumulus cells, like you see here. Cumulus cells will make good factors for the oocyte. This has been engineered, and so the groups in Melbourne have made a GDF9 with some parts of BMP15 so that we have a very stable molecule there that is called cumulin. We call it super GDF9, but actually it has the same function as cumulin. And so it will lead to, like it has been shown by 1 or 2 publications, to better embryo quality, to better blastocyst formation. And we have here studies that were done in our group. It's still submitted for human reproduction, where we see that if we add it in the capacitation system, we see that there is less apoptosis in the cumulus cells and that the nuclear configuration is increasing. So we have an effect on the condensation of the chromatin. And when you add it in the second part, in the IVM part, we see that the mitochondrial activity becomes higher, that you have more endoplasmic reticulum, and also cortical granules will be more present. There is no more reactive oxygen species or glutathione. So we have clearly shown that this molecule is very active, and that could lead to better embryo quality. So the future is ways to further improve the CAPA-IVM system. I think we have to define optimal day in a woman's cycle for the oocyte pickup without pre-treatment with hormones. We have to improve the needle for an atraumatic oocyte pickup. I think the needle that we have today is not easy. I think we can make something better. We have to implement oocyte secretion factors and other important cytokines in the media. We have to automate the IVM culture because it will solve the volume and it will save time. And also we have to optimize the vitrification of immature oocyte stages. Finally, I would like to thank very much my collaborators in Japan and in Vietnam, and also Rob Gilchrist. So here is Rob. It was when we visited for the first time the group in Vietnam. Is Dr. Lan, is Dr. Tong present here, and their famous group. And here we have the long-standing collaboration with the group of Yoshi and Isaac and Dr. Hashimoto here. And then also my collaborators in Peru. They are now back in their country. And also my group at the VUB, Ellen and Michel de Vos. I thank you for your attention.
Thank you, Johan. Fantastic presentation. 質問はですね、2人が終わってからにしますので、まずヨハン先生のですね、基礎的な以前の IVM、CAPA-IVM、そして将来の展望ということでお話しいただきました。Thank you, Johan. それでは次にですね、トン先生にベトナムから来られましたトン先生ですけれども、トン先生はですね、最初の IBM は実は IVF 大阪クリニックに学びに来られまして、最初は僕らが教えたんですけども、今はですね、きっと一施設としては世界最多の IBM、年間600件ぐらいをされております。ですごいたくさんの臨床例お持ちですので、トン先生の話を聞いてですね、それで最後にお二人にご質問いただけたらと思います。Dr. Tong, please.
Thank you for the introduction. And first of all, I would like to thank the organizing committee to give me an opportunity to present our experience in the application of CAPA-IVM in human practice. And I also would like to thank RipoLife to invite me here to give my talk. So this is our experience in the last 8 years, improving an application of CAPA-IVM system in our practice. This is where I'm working, Duc Hospital in Ho Chi Minh City. Actually, we went to Osaka to learn how to do IVM, and then we started IVMs in 2006. We have had more than 4,000 IVM cycles. About 2,000 IVM children have been born in Vietnam. After that, we try standard IVM without SCG. And with the collaboration with the JOHAN group in Brussels, we try to apply CAPA-IVM in our system in 2016. The first CAPA-IVM baby was born in our center in 2017. And up to now, we have had more than 2,000 cycles of the new CAPA-IVM protocol. And we have had more than 800 children have been born with the new technology. And actually, in order to have a number of mature sites to do IVF, we have two ways. First, we use stimulation, hyperstimulation, and we collect oocyte from the big follicles to get mature oocyte. And the second way is to collect oocyte from small follicles, and we mature it in vitro to get mature oocyte. And we are used to the first way with stimulation. And stimulation is not with the incidence of OSSS. We also have many problems with stimulation. We spend about 2 weeks for stimulation, actually just in order to get mature oocyte. IVF, it just takes part on only a few days. So we have complication of large fragile ovaries, venous thromboembolism. This is a picture show that a woman, in order to get a baby, he had to inject hundreds of injections. And this is a cost of medication and emotional burden to the woman. And we also have a long-term effect, adverse effect of stimulation, and it's very inconvenient for the couples to have IVF treatment. So that's why IVM has been quite attractive to our community. In the medical literature, we have 3 major IVM protocols have been practiced. The first is a standard IVM. It was assessed in 1991 by Cha and his team in Korea. The second IVM protocol is HGG prime IVM protocol. It is initiated by Tan Ayan in Canada, published in 1999. And we published the first live birth of CAPA-IVM in 2020. This is BIFASIC or CAPA-IVM. So this is the 2 old IVM protocols have been practiced in the world. And the problems of these 2 old IVM protocols are low maturation rate, poor oocyte quality, low number of embryos, and poor embryo qualities. That was why IVM could not apply widen in the world. And we try to apply the new CAPA-IVM, the 2 phases of culture IVM, to improve the IVM system. And actually, we add one more step of culture, as presented in Professor Johan Smith presentation. This pre-IVM step is to maintain the meiotic arrest in the oocyte cumulus complex. We maintain the transjonal projection for 24 hours to improve the oocyte development before we mature it genetically in other environment. By the combination of 2 steps of culture, we can improve the embryo development and pregnancy rate. And this is our protocol for CAPA-IVM in our hospital. At the beginning, we give 2 or 3 day FSS, no SCG. We collect the GBO side, CAPA culture for 24 hours, and IVM culture for 30 hours. We get mature side and we produce embryos and we transfer. First baby was born in 2017. The difference in the ovum pickup techniques are shown here. This is with the conventional stimulated IVF, and this is with the IVM OPU. The timing is different. This is 42 hours after the last FSS injection. Or if we do not give FSS, actually we can collect the oocyte at any time. if the largest follicle is not larger than 10 mm. The ovaries are different. It is stimulated ovaries, it is normal ovaries. We use different kind of needles and different technique of aspiration. We can use, some center can use heparin because we have some blood clot in this technique of pickup. And we spend more time because we mostly apply it for PCOS women. They have a lot of follicles, and it's a little bit more difficult to collect the immature aside from small follicles. So this is our CAPA-IVM system, and this is how we allocate time allocation in our daily practice, because we do it together with our conventional IVF program at the same time. So that is how we arrange time that can go very well with the normal IVF centers. So we will collect oocyte in the morning after 42 to 46 hours of the last APS injection, if we inject APSs in the morning. This is the GABA culture for 24 hours. And this is the composition of the media for IVM cultures. And after that, we do ICSI to produce embryo, and we grow to day 3 or day 5 embryos before transfer in the next cycle. I would like to show our results so far during the last 8 years. This is an initial small trial. In the first 3 small trials, we show that the CAPA-IVM system can improve the maturation rate and the percentage of good quality embryo on day 3. And in the last study, we show that CAPA-IVM can improve the clinical pregnancy rate and live birth rate compared with standard IVM. After that, we conducted a large trial, compare IVM and stimulated IVF in PCOS women or women who have high AFC. The paper was published in Human Reproduction in 2020, 4 years ago. We recruited 546 women with PCOS or high AFC. We compare IVM, CAPA-IVM, and stimulated IVF antagonist protocol. At that time, we still use day 3 transfer because our system was not very good at the beginning. We transfer in day 3. Most of them were 2 day 3 embryo transfer. And we found that the live birth rate after the first transfer are comparable between the 2 groups. And they have similar obstetric and perinatal outcome at birth. The children were born after the 2 strategies. Recently, we published a new large trial. We compare CAPA-IVM with FSS priming and without FSS priming. Recently, we improved our culture system, so now we can grow to proptosis for embryo generated from CAPA-IVM. We would like to know if we use 2 or 3 days of FSS before IVM pickup will provide different result with CAPA-IVM without any hormone injection before pickup. And this is a large trial prospectively registered in the internet. And this is what we did with our intervention in this study. The first group of patients, we give 2 days of FSS, and 42 hours we do egg pickup, 2 phases of culture. We do ICSI, and we grow embryo all the way to proptosis, and we transfer only proptosis. For this, the study group, we give no FSS priming. The patient just go directly to egg pickup, 2 phases of culture, ICSI and proptosis culture, and we transfer only proptosis. And this is the results of the 2 study group. This is non-FSS. We have 60 patients with non-FSS and 60 with FSS. The number of COC recruited, the mature oocyte, the fertilized oocyte, day 3 proptosis, good proptosis, and frozen proptosis were not different between the two groups. And this is the pregnancy outcome. We transfer mostly one proptosis for each patient, 85.5% in the non-FSS and 88% of the patient in the FSS group will transfer only one proptosis, and some of them will transfer 2 proptosis. And the clinical pregnancy rate, the live birth rate, were not different between the two groups. That means we do not need FSH priming before CAPA-IVM. Based on that study, that is our current protocol for CAPA-IVM for PCOS women who have no regular cycle. They can come to our clinic. We have a scan. If we found good number of follicles less than 10 mm, we can go directly to ovum pickup to phase of culture, do ICSI and grow proptosis. So our CAPA-IVM protocol now for PCOS women is no hormone injection. And actually, because they have no cycle, they can have ovum pickup at any days in a week. They can choose the day of pickup if they want. And easiest for the patient. So IVF is now just one pickup and then they go home waiting for the result of embryo culture. And then we can arrange for embryo replacement later. This is the data show the safety of our technique. After the large RCT published in 2020, we follow the children of the two groups, CAPA-IVM and IVF, for at least 2 years. And we found that the development of children between CAPA-IVM and IVF were not different. There's another paper we published in Journal of Assisted Reproduction and Genetics. We compare the development of children born from CAPA-IVM and children born from natural conception who were born at the same period of time in our hospital. And we found that the development of CAPA-IVM children were not different from the normal children. And recently we published an article. We did the epigenetic analysis between the children who were born with CAPA-IVM and children and women with normal IVF protocol. And we found the epigenetic analysis was not very much different between the two groups. So it means our technique is same, at least in the medium term. So what are the indications for CAPA-IVM now? The major indications are women with PCO or PCOS, those who have high risk of OSSS. And actually, these kind of women are good prognosis. They don't need to have many embryos. And I think CAPA-IVM is good enough for them to get several embryos for transfer. And we also can apply this for elective egg freezing. This is the case we reported in Journal of Assisted Reproduction and Genetics recently. This was a 30-year-old woman. She came before marriage, would like to have her egg frozen. We did 3 cycles of pickup without any hormone injection. And the first cycle, she got 20 eggs. Second cycle, 20 eggs, and third cycle, 14 eggs without stimulation. After that, she married and come back, and she want to have baby, want to do IVF. We warm this 10 mature oocyte, 6 M2 and 4 M1 oocyte. And then we fertilize and grow 2 proptosis. We transfer 1 proptosis. She got pregnant and gave birth. And the survival rate of vitrification for mature oocyte after CAPA-IVM is 10 on 10, 100%. And she still have 11 M2 and 7 M1 frozen at our hospital. So it can also work with elective egg freezing. And this is another indication for IVM. is a resistant ovary syndrome. It's a rare condition where the follicle don't respond to FSH at all. So this woman have no ovulation, even she still have small follicles in her ovaries. And we reported 2 cases live birth in 2021. And actually we have applied IVM for 6 cases, and all of them have pregnancy successful. So this is a very promising application for IVM. Without IVM, this woman have to go for egg donation because we could not stimulate them. To collect immature oocytes and mature it with CAPA-IVM from the ovarian tissues. These are the publications in Europe show that we can collect the oocyte from the tissues and mature it in vitro with standard IVM or with CAPA-IVM. And we tried to apply it in our program recently. After the woman have all the ovary excised, we collect the tissues and try to recruit oocyte from the tissues. And it is our protocol recently. We use needles to collect the oocyte from the tissues, and then we slide it before freezing. We try to collect oocyte, and we vitrify the oocyte as well. So those women who have indication for fertility preservation, they can collect their tissue for vitrification and also the oocyte, and then mature it for oocyte vitrification. And this is the oocytes we collect from the tissues. After the 2 phases of culture, we can produce mature oocyte, and we freeze it. This is our initial experience with CAPA-IVM with the immature oocyte from ovarian tissues. This is our learning curve. You see the last case, we have more oocyte and good maturation rate. So this is the oocyte we vitrify for each cases. And this is the guideline, the European guideline for female fertility preservation. Actually, we can collect immature oocyte from the ovaries in the body, or we can collect the oocyte and mature it from ovarian tissues. This is recommended by the ASRA. And in 2021, ASIM issues a committee opinion consider IVM a standard treatment, and with the potential indication includes high-risk OSSS, PCOS women, or fertility preservation. The very recent guideline, international PCOS guideline, recommend IVM as a first-line treatment if available before stimulation and IVF ICSI. So nowadays we have few indications for IVM, especially for CAPA-IVM. In our system now, CAPA-IVM is the first-line treatment for PCOS women who require ART treatment. And we can provide CAPA-IVM for women who would like to have fertility preservation. And we also apply CAPA-IVM for other group of patients, including gonadotropin-resistant ovary syndrome, elective egg freezing, patients who require patient-friendly ART, where they don't want to have hormone injection. And many of them we can apply CAPA-IVM. And those who have good ovarian reserve, young patients, they don't need to have many embryos to be pregnant. So we can provide CAPA-IVM with a very simple treatment. And for those who have very low ovarian reserve, what kind of stimulation you have, they still only have 1 or 2 embryos. So we can have the same number of embryos with CAPA-IVM without any stimulation. So in summary, the new IVM protocol is effective, and it can improve oocyte maturation rate, egg quality, embryo quality, and implantation rate. A hormone-free ART with single proptosis transfer is feasible and efficient for some group of patients. With the new IVM technique, we need to have some additional techniques, including oocyte pickup with small follicles, how to search and retrieve small COC from bloody follicle fluid, extra culture step and additional culture media, more labor in the laboratory. Yet, we do not need any additional equipment. Safety has been assured and future improvements are expected and studied. We can extend CAPA-IVM for another indications like young good prognosis patient, fertility preservation, and for elective egg freezing. This is the photo that we first met with the group from Brussels and Australia when we started the new protocol in Ho Chi Minh City 8 years ago, 2016. Thank you for your attention.
Thank you, Don. トン先生からはですね、IVM、CAPA-IVM のその臨床の有効性、それから、生まれた子どもの安全性ですよね、それから卵子凍結への応用、それから IVF 困難症例に対する応用とか、それから卵巣組織から、いわゆる妊用性温存に至るまでですね、広い範囲の話をしていただきました。それではですね、お二人に質問のある方はですね、会場の方、日本では IVM あまりされてないんですけど、会場の方、はい、どうぞ。
I'm Shotaro Higuchi from Shinshu University. Thank you for your lecture. Previously, we succeeded live birth by HCG primed IVM from ovarian tissue oocyte. We recently performed CAPA-IVM on a patient with ovarian fibromatosis with PCOS using ovarian tissue oocyte. We confirmed her pregnancy last week. We referred to your protocol. But since the granulosa cell was so few, we selected DB cyclic MP instead of CNP. Could you explain why you think CNP might be better?
See, I explained you, or I said it previously, there is no need to use pharmacological factors in media. I mean, it's much easier to get approval from regulatory agencies if you use natural factors that are already present, you know. So but I agree that it's not because you have used dibutyryl cyclic AMP that it cannot work. I think I've never said that. Now, why is it better? Probably I think it is following the nature. I mean, normally in a normal in vivo follicle, it's also CNP that goes via the receptor and that generates cyclic GMP, inhibits the PDE3, etc. It's a normal, it's following the nature, actually. That's what we try to do, is to following what nature has dictated, trying not to involve pharmacological factors if we can avoid them. That's my point. But if you ask me a biochemical mechanism, no, we have not yet clarified it. It's still a lot of research that can happen around that thematic.
それ以外にご質問、日本語でももちろん OK です。どうぞ。どうぞ。
日本語でも大丈夫なんでしょうか。高木病院女性医療センターの北島と申します。スミッツ先生にお伺いしたいんですが、カルチャーメディウムにエストラダイオールを入れることが非常に重要とおっしゃってたんですけれども、それはその生体内、先ほどネイチャーというお話をされましたが、ネイチャーでのフォリクルディベロップメントにおいてもエストラダイオールというのはヒューマンのオバリーにもインディスペンサブルなんでしょうか。
No. I think that estradiol is very important in the ovary. It has been clearly shown by the group of EPIC and also by Chinese or Japanese paper that if you do not, first of all, in the normal follicle, if you measure estradiol, it is 1,000 times higher than in the blood of the woman. 1,000 times higher. Okay, so it has been shown that if you add at least 10 nanomolar in your medium, that the expression of the NPR2 receptor, which is the receptor for CNP, will be expressed fully. And so you need the receptor in order to have an activity. And it's not different from nature. It's replication from nature.
So it means that there's a possibility to adding the estradiol to the follicle stimulation.
No. It's not needed during stimulation because you have enough estradiol produced by granulosa cells in a follicle. Yeah.
I understand. Yeah. But there are some women with deficiency to produce the estradiol, but the follicle matured. They pick up the oocyte and they get pregnant. Are there any explanation for that?
It's a good question. If you say deficient, you think it is 0 or is it lower than, because in a normal follicle it's 1,000 times higher than in the blood. So you need, I think estradiol is everywhere in the body of a person.
Thank you very much.
Yeah.
それ以外、なんか、どうぞ先生、国立先生。
Thank you very much for wonderful your lecture. We are going to try CAPA-IVM advised by Dr. Morimoto. And clinically, how many eggs are typically retrieved on average? Please tell us.
トン先生でいいですか。
はい。
Dr. Tong.
Yeah, when we apply CAPA-IVM mostly for PCOS women, then with the PCOS woman, the number of oocytes we get for each pickup is an average from 15 to 20.
50 to 20. Wow.
And the maturation rate with CAPA-IVM is about an average of 65%. So we get enough mature oocyte for IVF and ICSI. And actually the largest number of oocyte we get from one patient is more than 70. And we can make more than 20 plastocysts without any stimulation. It depends on patient.
And next question, and please tell us what is the exclusive case? For example, hypogonadism and hypogonadism is okay for CAPA-IVM, hypotype case.
Yeah, I think it depends on if we can find antral follicle in the ovaries, we can collect the oocyte and mature it. So it depends on what we have in two ovaries. If we found follicles, we can collect immature oocyte and mature it.
Thank you very much.
それ以外、ございますでしょう。吉田先生どうぞ。
Thank you very much, Johan, and the very nice and excellent lecture for us. I have a question that is in the case of the PCO case, some patient has already in the antral follicle size is sometimes over 8 millimeter like this. So how do you think of the CAPA-IVM in these cases?
Over 8, you mean? You mean
8
what?
millimeter.
8 millimeter. I
And
think
they have lots of antral follicle, and then in that size, it's a winner doing the echograph, sonograph, echograph, and then the dominant, like a dominant follicle, it's it's many of the antral follicle are there. The size, the maximum size is over 18 millimeter. How do you do all of the Kappa-IVM? It does the collectively the doing of the direct the aspiration or not?
I think that there is no difference in aspiration, but Dr. Tong has done much more cases than we have ever done in Brussels, right. So maybe he has another opinion on that.
In the past, when we use HCG IVM, epicenter priming, and HCG, we found that it's easier to puncture the large follicles, and we have more oocyte. But when we changed to no HCG and Kappa-IVM, we found that we have better result with follicles about 6 mm or less. It's easier to collect the oocyte without HCG with the smaller follicle than the large follicle. The follicle that is more than 6 to 10 mm, the recovery rate without HCG, it seems to be lower than the small follicles. Yeah.
Okay, thank you very much.
では小山先生どうぞ。これ最後の質問になります。
ちょっと分かりやすい質問を日本語で皆さん分かるようにお願いします。Kappa-IVM ですね、これの一つの理想形、未来の理想形というのは、卵巣刺激のない IVF 治療の実現だと思うんですね。で、現在例えば普通のブラストシトトランスファーで 40% の妊娠があると、100% になるためには3つの胚盤法が必要です。ではこの3つの胚盤法を作るために今のキャパのシステムで何個のスモールフォリカルの COC があればこれが実現できますか？
I'm talking about the data that we have in Vietnam. I think on average, if you have like 10 to 12 metaphase 2 oocytes, starting from 20 COC, you can have 2.8 plus minus 2 blastocysts. Yes. So that's what the data show from the last study that Dr. Tong mentioned.
Yeah. From the recent paper we published in Fertility and Sterility, we grow to blastocysts for all women. The average number of COC we got from one patient is from 16 to 18. Oh. And we will have 2 to 3 blastocysts at the end. This is for women with PCOS. But for those with not PCOS, just a PCO-like many high ovarian reserve who have about more or less 20 follicles at the beginning of the cycle, usually we get less number of oocytes, less number of embryos. But I think we can still have room to improve the system in the future in terms of the ovum pickup technique and also the culture system. I think we have more room to improve it in coming years.
Yes. Thank you. Please do it. Thank you.
Thank you, Johan and Tom. 本日はですね、Kappa-IVM ということで、リプロライフの共催でこのセッションを、セミナー、開催させていただきました。IVM ですね、非常に採卵が難しいというのはありますけれども、今、トン先生言ってましたように、実は小さい方が取りやすいんです。日本で IVM やってるのは、メインには私、IVF 大阪と仙台アートしか今のところあまりないと思うんですけども、我々のところは、いわゆるルーチンにIVM をしておりますけれども、 卵のサイズはですね、大きい方が取りやすいと思ってですね、10ミリとかそれ以上にするんですけど、実はあまり取れないです。それにマチュレーションレートも良くないです。5ミリ、6ミリの卵胞で十分に選子できます。で、トン先生のところはですね、50個から20個って言われてましたけれども、我々のところでも最高40個以上、1人の患者さんから取ったことがあります。やはり日本人は PCO のパーセンテージが低いと思うんですね。ベトナムとかに比べては。ですけれども、IBM で我々のところも150人ぐらい赤ちゃん生まれてますし、十分に臨床応用可能な技術だと思います。で、もともとはヨハン先生から習った技術なんですけれども、やはり OHSS がない、0% です。で、今ちょっと保険でですね、なかなか難しい面もあるかと思うんですけれども、AMH が10以上の人であれば、我々のとこ施設では、ファーストチョイスは IBM です。やはり OHSS を避けるということで、それと保険でも、移植が回数制限に引っかかりますので、今はブラストシストかなりいい確率でできると言われておりましたけれども、ブラストができない場合には保険の回数は減りませんので、AMH の高い方には危険を回避するという意味で、私はIVM 今後ともですね、CAPA で効率が良くなればますます必要になるんではないかと思います。やはり安全が医療の第一ですから。本日はですね、共催セミナー、リプロライフと共催で、CAPA-IVM ということで、開かせていただきました。ご参加どうもありがとうございました。